All right, welcome to The Explainer. Today we're diving into what's basically a microscopic true crime story. We're tracking one of the most highly evolved, deadly microscopic invaders in human history. It's an absolute master of disguise, a ruthless hijacker of human cells, and the cause of a disease that has quite literally shaped our evolution and history: malaria. Whether you're a medical student, an avid traveler, or just someone fascinated by biology, understanding how this pathogen operates is absolutely crucial.
Okay, let's map out exactly what we're covering today. First up, we'll meet the Plasmodium parasite. Second, we'll trace its complex life cycle. Third, we're looking at the different species and the symptoms they cause. Fourth, we'll dive into diagnostics. And finally, we'll wrap up with prevention and future vaccines. Let's get right into it.
Section 1. Meet the parasite. Plasmodium biology.
Over 1 million. Take a second to really think about that number. That's how many children in Africa die from this disease every single year. And that's out of hundreds of millions of global cases. So this isn't just some abstract microscopic entity. We're talking about a pathogen that takes a massive, truly tragic toll on human life, mostly in poorer tropical areas of Africa, Asia, and Latin America. This is exactly why understanding this matters so much.
Now, what's really interesting about what we're looking at here is the parasite's anatomical weaponry. Under an electron microscope, you can see this complicated anterior end. It's called the apical complex. And guys, this is quite literally a microscopic breaching kit. You got these structures called rhoptries, polar rings, and micronemes. They're not just biological decorations, they're highly specialized tools the parasite uses to latch right onto human host cells and forcefully break its way in. It's wild.
So, because of that intense little breaching kit, Plasmodium is grouped into the Phylum Apicomplexa. It actually shares a family tree with other notorious parasites you might have heard of, like Cryptosporidium or Toxoplasma. But here's the crazy part. They don't just live in one host. They literally bounce back and forth between sexual and asexual reproductive phases. It's this continuous alternation between an arthropod, like an insect, and a vertebrate, like us humans.
Moving on to Section 2. The life cycle. From mosquito to human.
Okay, so we know it has the weapons, right? But how does it actually breach our bodies in the first place? Well, it all starts with the delivery mechanism, the blood-feeding female Anopheles mosquito. And a quick note here, it's only the females that bite. They're the ones that need the protein from our blood to develop their eggs. So when an infected female takes a blood meal, she inadvertently inoculates the human host, basically injecting the Plasmodium parasite right through her saliva.
And this invasion, it's not random. It happens in a highly structured three-step sequence. Step 1, the mosquito inoculation we just talked about, introducing the parasite. Step 2, the parasite goes into hiding and multiplies. We call this the Exoerythrocytic stage, which just means outside the red blood cells. Specifically, it sets up camp in the human liver. And finally, step 3, the erythrocytic stage. This is where it bursts out of the liver to systematically invade and destroy human red blood cells. It's a calculated attack. Let's trace the actual flow of this invasion. So starting from the moment of the bite, the mosquito injects parasites in a specific form called sporozoites. Think of these like little heat-seeking missiles heading straight to the liver. Once they're inside the liver cells, they develop into dividing forms known as schizonts. Then, bam, they release thousands of merozoites. The merozoites are the form that bursts out of the liver and goes hunting for your red blood cells. Once they're inside the blood cells, they feed, they grow into trophozoites, they multiply again into more schizonts, and then they literally burst the red blood cell wide open to infect even more cells. It just cascades. And eventually some of them develop into sexual stages, gametocytes, just waiting in the blood to be ingested by the next mosquito so they can start the whole shebang all over again.
All right, let's move to Section 3. Species and symptoms, clinical manifestations.
Now that vicious cycle of bursting red blood cells, that's the direct cause of the physical devastation the human host experiences. But here's the thing, not all malaria is the same. Take a look at that top row, Plasmodium falciparum. This is the malignant, highly dangerous variant that's actually responsible for the vast majority of malaria deaths. It triggers this acute fever every 48 hours, or sometimes just an unpredictable acyclic fever. Contrast that with the bottom row, Plasmodium malariae. That one has a 72-hour quartan fever cycle, and terrifyingly, it can linger in your body for up to 20 years. Meanwhile, you've got vivax and ovale sitting in the middle, and those guys can actually hide out dormant in the liver and cause a relapse way down the line. So, as you can probably guess, the clinical damage we see in a patient is inextricably linked to that microscopic destruction of red blood cells we just traced. Imagine millions of your red blood cells bursting open simultaneously. The body's reaction is intense. Cycles of high fever,
shaking chills and drenching sweats. And because so many red blood cells are just gone, the patient suffers from severe anemia. In the absolute worst cases, especially with that falciparum strain we mentioned, you see horrific complications like blackwater fever, or even cerebral malaria, where infected red blood cells physically stick together and block the blood vessels supplying the brain. It's incredibly dangerous and often fatal.
So, the crucial point here, and this is a major point of confusion for a lot of medical students, is understanding how malaria can sometimes return after a patient seems totally cured. There's a very precise biological difference between a relapse and a recrudescence. A relapse is caused by those dormant parasites in the liver, they're called hypnozoites, waking up weeks or even years later. And remember, that only happens with the vivax and ovale species. Recrudescence, on the other hand, is a failure to completely clear the blood stage infection. This is often because of drug resistance or a weak immune response. The parasites basically just rebuild their numbers. This distinction is absolutely huge in the real world. If a patient gets sick again, doctors really need to know, did dormant liver parasites just wake up, or did our medication just fail to clear the blood? It completely changes how you treat them.
Moving right along to Section 4, Diagnostics, Clinical Testing.
So, how do medical professionals actually track down and identify this microscopic invader in the clinic? Well, the gold standard for decades has been microscopy. Now you'll see two types here. First, the thick blood film. For this method, the red blood cells are actually intentionally burst open. It concentrates all the blood elements into one spot, making it a fantastic, super fast screening test just to say yes, the parasite is definitely there, and to measure the density of the infection. But then you have the thin film. Here, the red blood cells are fixed in a single, undisturbed layer. Now, this takes a lot more time to read, but because it leaves the cells intact, the microscopist can identify the exact species of Plasmodium based on how the parasite physically looks inside the cell.
But here's the catch with microscopy. You need a lab, you need electricity, and you need highly trained staff. And in a lot of affected areas, you just don't have that. Enter Rapid Diagnostic Tests, or RDTs. This was a real game changer. Instead of hunting for the parasite visually, these tests hunt for chemical fingerprints, specific antigens that the parasite leaves behind. A major one to know is histidine-rich protein 2, or HRP2, which is produced exclusively by that deadly Plasmodium falciparum. By detecting these proteins, RDTs make a rapid field diagnosis possible in minutes. You don't even need a lab, you can literally do it in a remote village.
But let's objectively weigh the pros and cons of these RDTs for a second. The advantages are obvious. No lab, no electricity, and you can train a healthcare worker to use them accurately in literally one day. But they aren't perfect. For one, the test strips can be completely ruined by extreme heat or humidity. Secondly, unlike a thick blood film, they can't tell you the intensity of the infection. And crucially, this is a big one. Because they detect proteins that can linger in the bloodstream, a patient who has been successfully completely treated might still test positive weeks later. It's a pretty major blind spot.
All right, final section, Section 5, Prevention, Future Strategies.
Long before we even start talking about vaccines, humanity has relied on a massive, multi-pronged strategy called vector control. The idea here is simple. You don't just attack the parasite, you go after the mosquito. This means widespread use of insecticide-treated bed nets to protect people while they're sleeping. It means taking prophylactic drugs to stop the infection before it can even take hold. And it means serious environmental engineering. We're talking draining standing water, spraying larvicides, and even using biological warfare. Yeah, biological control. For example, introducing a specific species of fish, the gambusia, into waterways specifically because they love to snack on mosquito larvae. It's all about breaking that breeding cycle.
Of course, the ultimate weapon would be a vaccine. And scientists right now are attacking this on 3 completely different fronts, targeting different life cycle stages. The first frontier is the anti-sporozoite vaccine. This is designed to neutralize the parasite the absolute second it enters the blood from the mosquito bite, totally preventing the initial infection. The second strategy is transmission blocking. Now this is fascinating. It's actually designed to arrest the parasite's development inside the mosquito itself, essentially stopping the disease from being passed on to the next person. And the third frontier is the anti-asexual blood stage vaccine. This one doesn't stop the infection itself, but it's designed to dramatically reduce the severe clinical symptoms to prevent death, which is absolutely crucial for protecting high-risk children.
So what does this all mean for the future? It brings us to the ultimate question. Which vaccine strategy, blocking the initial sporozoite invasion, stopping that devastating blood stage damage, or breaking the mosquito transmission cycle entirely, will finally be the key to eradicating this ancient disease? It's literally one of the greatest biological challenges of our time, but as we've seen today, the ingenuity of the scientific response is just as fascinating as the complexity of the parasite itself. Thank you guys so much for joining me for this explainer, and keep questioning the science around you.